Good afternoon, everyone. I'm Diane Tuff, a board member for Exposomics and the Children's Environmental Health at Mount Sinai. Thank you for joining us for our last webinar in our Environment and Health Lunchtime chat series. I'm really excited for today's discussion with Dr. Douglas Walker, who I will introduce shortly. If you've turned into earlier talks, you've learned about the new research that's coming out of Mount Sinai on a variety of pollutants in our environment and how they affect human health. As a photographer and filmmaker who has traveled to some of the most secluded places in the world, including the Arctic Circle, places that we would expect, or at least hope, would be pristine and untouched. I know firsthand how the warming climate is changing these parts of our Earth, but it isn't just climate. Environmental debris, including microplastics, have been found in the Arctic Sea and the deep ocean floor. How this phenomenon is affecting our Earth is a talk for another time. Today, we're going to hear about how it is affecting our health and hopefully what we can do to prevent exposures for ourselves and future generations. Our challenge scientists like Dr. Walker face is that it isn't just how plastics affect health, it's how do the thousands of related compounds in plastics affect health. This complex and situation requires new approaches. Today, I'm hoping Dr. Walker will share with us how Exposomic framework is making this possible. Today's presentation will be about 25 minutes, followed by 20 minutes of questions and answers. Please note that the chat is disabled, so please use the question and answer function on your computer, and we'll try to get as many questions as possible. It's my pleasure now to introduce Dr. Douglas Walker. He's assistant professor in the Department of Environmental Medicine at the Public Health and a member of Mount Sinai Institute for Exposomic Research. Dr. Walker joined Mount Sinai in 2018 from Emory University, where he completed postdoctoral training, and he received his PhD in 2017 from Tufts University in environmental and water research engineering. Dr. Walker, please take the stage.
Great. Thank you so much for that introduction. And I would like to thank the organizers for the opportunity to share some of the research we're doing in the Institute for Exposomic Research on developing new methods that allow us to understand complex plastic exposures in human populations. So it should come as no surprise to all of us that global plastic production has steadily increased since the 1950s to support many of the common items and products that we use in our daily lives. Since the 1950s, we've seen approximately 8 billion metric tons of plastics produced, with the annual plastic production steadily increasing each year. This does not account for the rapid increase in production of many single-use plastic materials that have occurred due to the ongoing COVID pandemic. Unfortunately, the current use trends for many of these plastic materials are single use, meaning they're used once for their primary purpose and then either placed in landfills or recycled for additional use. Of the approximately 8 billion metric tons of plastics that have been produced, about 2/3 of those end up in landfills. Approximately 5 to 10% is recycled, and then after a second use, placed in landfills, and only about 1/3 of those are used more than once. So this is a tremendous environmental and ecological burden that we face from plastic materials. And a lot of what concerns me and the collaborators that I'm working with about these plastic materials is what happens to them once they are released into the environment. Many plastic materials undergo a number of different weathering processes that can change or interact with the plastic material itself. And this can result in very complex mixtures of plastics that provide a very significant human and ecological exposure burden. While there has been a tremendous amount of research studying plastic exposure in human populations, most of those are considering plastic-related chemicals and additives that leak from plastic materials. What we're starting to recognize is that additional plastic exposures arise from plastic particles that are formed through these weathering and use patterns. And in fact, exposure to these plastic particles represents a very large amount of the exposure burden that we experience. So where do these microplastics come from? Microplastics can originate from a very large variety of plastic materials. Oftentimes they can be produced just from the typical use patterns that we're applying to our plastic material. For example, many of the plastic bottles and baby bottles that we use actually shed microplastic and nanoplastic particles. Oftentimes we find these plastic particles in a number of other products, such as cosmetics, paint, and other commercial formulations, and also our tires themselves weather as we're driving our cars, and these release what's considered microparticles or microplastics in the environment. So these microplastics are represent a very, very large burden of our plastic exposures. When these particles form, there's no uniform process that creates them, and the different weathering processes can result in different types of microplastics. For example, some can be present in beads that are in different soaps and other materials. Probably one of the most common types of microplastics that we see are from fragments. And again, these are from the weathering or physical processes that are breaking down plastics. Another large burden is fibers. These can come from a number of different sources, such as our clothes and dryer lint. And there's also a lot of recent concern about our potential increase in exposure to fibers from the masks that we're wearing due to COVID. These microplastics have been shown to enter your body through a number of different pathways. They're present in much of the food that we eat. They've been found in our drinking water. We tend to breathe in the smaller nanoparticles and fibers. So there are multiple feasible routes for these microplastics to enter your body. And a lot of what interest in my research is focused on is what happens to these microplastics once they're in your body. Can we actually develop techniques that allow us to assess the presence of microplastics in different tissues and in populations, and also to understand how the presence of those microplastics could be affecting your health? Because the processes that are forming these micro and nanoplastic particles are so heterogeneous, there's a number of different sizes of particles that we can be exposed to, ranging from macro size particles, which we can see with our eye, through micron-sized particles, which are not visible to the human eye, but are roughly the size of a cell or a bacteria, or even the diameter of your hair. And then there's also nanoplastics, which are much, much smaller than bacteria and cells, and tend to include any plastic particles that are less than 100 nanometer in size. So these are very, very small particles. And what we're trying to understand is how these particles, particularly the different sizes, could potentially be taken up into your body, how they can pass into your bloodstream and into different tissues, and how these could be affecting your health. A lot of the research that's occurred in microplastic studies has used what's referred to as microscopy methods to measure microplastics. And this is where you use different microscopes to look for the presence of microplastics in your samples. These work very well for larger plastics, but unfortunately, the smaller nanoplastic particles are not visible most of the time using these methods. So we have almost no idea what the true size distribution are of the microplastics that we're exposed to. We're starting to recognize these exposures as a growing public health concern, and we're really starting to consider microplastics as an environmental pollutant of emerging concern. Microplastics have been found in what's considered pristine environmental areas where there's minimal or very little human interaction. And this suggests that these microplastics are being transported globally into even what we would consider very clean and very pristine environments. Initial studies have shown that microplastics can be found in human stool samples, suggesting again that we're intaking a relatively large amount of these. However, some of them are being removed from our bodies. There's been a number of studies showing that plastic fibers that are being inhaled can be found in different regions of the lung. And a study that just recently came out showed that the size of the particle influences the region of the lung that they're deposited in, with the smaller particle reaching deeper areas of the lung. There's been studies coming out showing that microplastics are very common in our pet food and can be measured in our pet feces and other waste products. Very concerningly, they've recently measured different microplastic types in both infant and adult feces. Obviously, many infants are using plastic bottles, and so there's a very high potential for exposure to microplastics through that. And just this month, from a group in the Netherlands, there was a publication showing out that came out showing we can measure these microplastic particles in human blood. So the evidence is suggesting that these are a very persistent and ongoing exposure that's widely experienced by many individuals and populations. The challenge we face is trying to identify ways to actually measure these exposures. Now, this is a very complex figure, and I'm not going to spend the time explaining what it all means in this, or what this means, but just a quick summary of it is that it's essentially showing how many different chemicals have been linked to plastics and plastic production. And this was some excellent work that was published from Helen Weisinger about a year ago, where she systematically went through many different industrial chemical databases to see how many compounds were associated with plastics. And the most important point to take from this figure is, one, that plastics are very heterogeneous and come from a number of different manufacturing sources. And two, is that there is a very, very large number of different compounds that are being used in plastic production. And that is what is represented by this number here, showing that using files for these different plastic products, she was able to identify over 10,000 unique compounds associated with plastics and plastic production. So when we think about developing methods that allow us to characterize exposure on this scale, it is not possible to develop traditional analytical methods that allow us to evaluate all of these exposures. We really need to think about cutting edge and new strategies that better allow us to characterize plastics to this extent. And this was highlighted by a recent workshop by the National Academy of Science that was held back in July of 2020, which was specifically focused on emerging technologies for research on microplastics. And one of the key summaries from this meeting was that in order to develop the critical measures that we need to understand microplastic exposures in humans and health effects, it's very important to take a cross-disciplinary approach that allows a collaboration across many different areas of environmental health sciences, human health, and polymer chemistry. And this, in a sense, is the concept of the exposome, where we're using many different strategies, both analytical and data-driven, to understand the complexity of the exposures that we experience in humans. So much of the work that's ongoing in my laboratory in the Institute for Exposomic Research is to use exposome scale analytical strategies that allow us to monitor the complexity of microplastic and plastic related chemical exposures in human samples. And in order to understand exposures on this scale, we need to apply measurement methods that allow us to detect as many different exposure biomarkers in a human sample as possible. And when I say human exposure, or when I say exposure biomarkers, what I mean is a specific chemical signal that we can detect that can allow us to predict an exposure that an individual experienced. This is important for two reasons. It allows us to understand how widespread exposures are across a population, and it also allows us to assess an exposure burden and see how that is related to different health outcomes. So to do this, we have to essentially take exposome or untargeted analytical strategies to develop the methods that we need to measure the plastic exposome. And what I mean by this is that we're using a different approach from traditional methods for assessing exposures. For traditional biomonitoring studies, which can be referred to as targeted exposure monitoring, you're developing assays that allow you to measure specific exposures. And this often allows you to characterize anywhere from 10 to 100 different chemical exposures. But as you can tell, this is much, much less than the approximately 10,000 different chemicals that are used in plastic production and materials and could be important exposures we experience from plastics. So to better measure the plastic exposome, we use untargeted methods that were developed to broadly detect as many different chemical signals in a biological sample as possible. So we're optimizing our methods to screen for tens of thousands, if not hundreds of thousands of signals, and then see which ones are plastics and how we can attribute those to the plastic exposome. And this is giving us the ability to now monitor for exposure to thousands, if not tens of thousands of chemicals. And so what are the advantages of when we're using these approaches? And as it was, I mentioned in the previous slide, when we're assessing exposure to known plastic biomarkers, we're often limited to assays that only allow us to characterize about 10 to 100 exposures. And this is much, much less than the 10,000 compounds that are used in plastic productions. So when we're using untargeted approaches, we're essentially allowed to look at a much fuller picture of the exposures that are occurring from plastics. This allows us to assess exposure to things like environmental microplastics, which are the particles themselves. We can screen for the presence of additives or impurities that are there from the manufacturing of the plastics, as well as co-transported environmental pollutants. And what that means is what we've been finding is that these microplastic particles can actually pick up other environmental pollutants when they are being transported and carry those to populations or provide additional exposures in addition to the plastic related chemicals. So what strategies are we using to actually measure these microplastics? And this is just a very, very brief overview of the different aspects of microplastics we're trying to characterize. I would be happy to discuss the technologies and the science behind them in depth at a later time. But first and foremost, many of our measurements are focused on plastic chemical concentrations as well as particle concentrations. So we are using very advanced instrumentation that we have available through the Institute for Exposomic Research to try and both measure known microplastics as well as screen for as many other microplastic and plastic related compounds as possible. With collaborators in the Netherlands, we're also evaluating how the shape of microplastics can influence both their uptake and health effects. So as I mentioned previously, microplastics can come in a very wide variety of shapes and sizes, especially in a very wide range of sizes. And so one thing we would like to understand is how the size of the microplastics influences both the health and the exposures we experience. Another area of interest is microplastic accumulation. So in many cases, these are discrete particles. And one thing we would like to understand is how they can enter and accumulate in different tissues. So what we want to know is when we're exposed to microplastics, do we excrete them very quickly? Are they removed from our bodies? Or do they enter and accumulate in some tissues relative to others? So for example, do we tend to have microplastic accumulation in our gut, or do we see it in adipose tissue? And finally, we're interested in co-exposures. So microplastics, as I mentioned, can carry a large number of other environmental pollutants on their surface. So do these act as essentially an exposure source to other pollutants that could be influencing our health? And it's this, because of the complexity of microplastics and their heterogeneity, this multi-modality approach to characterizing different aspects of microplastics is really very critical to understanding potential health effects to them. And so for the next few slides, I'm going to introduce the Aurora Project, which is a collaboration with a number of other universities in the European Union, where we're applying this multimodality analytical approach to develop a risk assessment framework for early life impacts of micro and nanoplastic exposure. There's been a tremendous amount of interest in understanding both endocrine and particle exposures to a fetus and how that can both influence the fetal health and outcomes, as well as maternal health and maternal outcomes during delivery and pregnancy and after. There's been initial evidence showing that particles from air pollution can enter and accumulate within the placenta. So our research is focused on measuring the distribution of microplastics in different fetal tissues as well as maternal blood. And when I say fetal tissue, we are interested in placenta, cord blood, the cord tissue, as well as the meconium, which is one of the first fecal samples from the newborn. So this is an international collaboration where we are the only US institution with funding from the Horizon 2020 project in the European Union to contribute to research on microplastics. And our project, which is focused on early life impacts, involves about 6 other European countries where we're all contributing to different aspects of understanding health effects of microplastic exposure in early life. Next, I'm just going to mention a few important aspects of this project that will allow us to better assess the health effects of microplastics. So first, one of our goals is to perform what we can call a deep characterization of microplastics in maternal and fetal samples. So our goal in this portion of the project is to create as much evidence and understanding of early life exposure to microplastics. We want to understand what sizes are present, their surface chemistry, the types of plastic present, how they accumulate in certain tissues, and really generate as much information on microplastic exposures in the early life as possible. And this is primarily being led by Florian Meyer, who's a professor at Utrecht University in the Netherlands. The second portion of the project, which is primarily focused at Mount Sinai and is being led by 2 of the fantastic junior scientists in my lab, Anna Robach, who's a postdoc, and Brooklyn McNeil, who's a research scientist, is focused on developing scalable exposure assessment methods so we can actually measure microplastic exposures in a population-wide manner. So can we collect samples from pregnant women and look at how microplastics are distributed across the population in hundreds, if not thousands of samples? And so we're really empowering microplastic exposure assessment through biomonitoring. And one of the third key components is a toxicological part of the study, where they are using a tiered approach to understand both endocrine and developmental toxicity of microplastics. And they're doing this through a very interesting framework that's essentially starting from the basics, where they're using very simple cell models to understand toxic effects of microplastics, through placental perfusion and developmental toxicity, where with placental perfusion, they're actually taking microplastics and perfusing them into a recently delivered placenta to understand how the microplastics change the biology in the placenta, as well as the developmental studies to see how microplastic exposure alters development in early life in multicellular organisms. And this effort's primarily being led at Utrecht University and the VU, which is in Amsterdam, by Hanna Duza, who's a fantastic postdoc, Mare and another Florian who are also working on this aspect of the project. But our goal is to really perform these different measurements in a framework that allows us to perform the first risk characterization and risk assessment of microplastic exposure. So we're combining both our exposure assessment findings as well as our hazard identification findings, which we get from the toxicological studies and EPI studies, to understand what the potential risk is of microplastic exposure. This is very poorly understood. There's been a lot of contradictory evidence suggesting whether microplastics could pose a risk to human health or could be inert. And so we hope to empower one of the first frameworks that allow us to perform this risk assessment in human studies. And we're also extending this to different models and different types of studies. And so we're in the initial stages of developing a project in collaboration with Tanya Alderetti, who's at the University of Colorado, that will allow us to evaluate how digested microplastics, which we're primarily exposed to through our food and water, influence the gut microbiome and could contribute to inflammation in the gut and potentially inflammatory diseases. And this will be a very interesting and novel application of an exposome by microbiome analysis to understand the effects of microplastic exposure. So I'd like to thank everybody for your time. I greatly appreciate being able to share our research on microplastics. I'd like to acknowledge the funding that is primarily supporting this work, which is almost exclusively from the European Union, the Institute for Exposomic Research at Mount Sinai, which has been a fantastic environment to develop these methods, and also my fantastic team, and especially Anna Roebuck and Brooklyn McNeil, who have been working on the microplastics project and developing these methods. If you're interested in learning more about the project and other microplastics projects in the EU, we have a fantastic website that's now live. So please either follow the URL or the QR code and read up on the project. Thank you all for your time, and I'm happy to take any questions.
Thank you so much. That was really, really interesting. And we have quite a few questions. Are there studies that have shown how long-term impacts on babies from ingesting microplastics? Could there be concerns about feeding babies with plastic bottles?
That's a fantastic question, and we really don't know at this point. That's been one of the really surprising aspects about entering this area of research is there's such a lack of information on this exposure itself. We really do not have any idea of potential health effects or outcomes linked to them. And that's what we're really hoping to begin answering with some of these research, but most importantly, developing the methods that will allow us to evaluate these exposures and see how they could be linked to outcomes or health effects. I would say we're at a very early stage where we're working on the methodological aspects that will eventually empower us to start asking those types of questions.
Mhm. Here's another really good one. You describe how your methods can examine 10,000, maybe even hundreds of thousands of chemicals. How do you determine which ones are more important than others? And can you describe the new technology that allows you to do this?
Yeah, that's another excellent question. So there's a few different ways that we can prioritize our most important signals, and it's really dependent on the objectives of the studies that we're working on. A lot of what we do now is what's referred to as discovery-based methods, where we're developing these untargeted assays and then asking biological questions or disease outcomes to prioritize which are the most important. So what this means is we're selecting our populations and we're developing our untargeted methods, and then we're applying them to the samples that we selected. We then use a pretty wide range of different biostatistic and bioinformatic techniques, and even starting to use some deep learning methods to prioritize which signals are the most important. A good example would be a study where we're looking at a group of individuals who have a disease and a group of healthy controls. So how we would structure the study is to analyze all of the samples from both cases and controls, and then look and see what is different between individuals who are healthy and may have the disease that we're interested in. That's one way of prioritizing our signals. Another is, really we're interested in exposure surveys. And so we try to identify as many compounds as we can, and then link those to other covariates that could be driving exposures, such as different environmental differences, environmental disparities, or environmental justice variables, and other factors.
Mm-hmm. Interesting. Okay. And a lot of questions are coming in about the opportunities for stronger regulations on policy changes. Can you comment on the role of work like yours is doing to evoke meaningful change?
That's a challenge, and I don't. That's one of the things that honestly keeps me up at night when I think about my work. Is we're putting all of this effort into identifying these exposures, but what if an exposure is so widespread that it becomes very challenging to actually reduce our exposure to them? What do we do then? I think as we've seen with plastics, where they've phased out things like BPA or other additives that are known to leach from the plastics and could have health effects. I think there is advantages of both advocacy and as well as regulations that could control what type of plastics are being generated. I think the most important thing we can do at this point, though, is just all be responsible with our plastic use. Try and avoid the use of single-use plastics when that's possible. Stop contributing to the burden of plastics in the environment and in the materials that we're using. I think that would be the way to make the most difference at this point. I think if there were to be any regulation, it would be many years from now, and it would require a much larger body of evidence, to show that there is true harm from these materials.
Mm-hmm. Do we know anything about how long the microplastics persist in the body after they're ingested or inhaled? And do we also know if these inhaled microplastics enter the bloodstream? Does your method give you any information about the route of entry of microplastics, ingestion, inhaling, etc.?
Yeah, so that's another fantastic question. And again, what's been very challenging about this field is it's basically in its infancy in terms of really developing these biomarkers in human samples. So we don't know much about uptake. We don't know much about elimination. And a lot of the methods at this point are focused on measuring them in the biological samples themselves to show exposure burdens. Once we have a handle on that and do understand how to assess these exposures, we can start designing the experiments that will give us information on route of exposures. There's been some efforts to look at microplastics in air samples. That's something that can be done easily. And then as another part of the Aurora project, which I didn't really describe in the presentation, we actually have an intervention study. We have individuals who use, have regular plastic use patterns, and we're going to collect samples from them and measure them over a certain amount of time. And then we're going to have an intervention where we're going to provide all of their food prepared in glass materials and try and eliminate their use of plastics to see if we actually measure a reduction in microplastic burdens, or if it stays the same, or how that changes. But again, at this point, we're at very, very early stages of this research. And so it's challenging to give definitive answers. Right now, we're trying to understand the problem, basically.
Mm-hmm. So another question is, plastic that's spread through the air contributing to air pollution? Does that have anything to do with that?
Yeah, absolutely. And when we measure air pollution, one of the things we look at are particulates, and plastics can either be present as compounds on other types of particulate matter, or they can be, plastics can be part of air pollution as well. If you look at your dryer filter and see the lint in it, that is essentially microplastic and fibers that would be suspended in the air that you could potentially breathe in. There's a lot of sources for plastic aerosols that are present. So absolutely, it can be a source of air pollution. I don't think it's received even nearly as much attention as, say, automotive emissions, because automotive emissions do provide a much higher exposure source, but it can absolutely contribute to air pollution.
Mm-hmm, mm-hmm. And what about the studies on maternal and fetal health in microplastics? I know you said quite a bit about that.
Yeah, so this is the data we're trying to generate at this point. And so we're trying to look at the presence of these microplastics in birth tissue. And then Hannah Duza, who's a postdoctoral student at Utrecht, has some very exciting or very interesting research findings showing how microplastics affect placenta cells. So she's running models in the lab where she takes different types of placenta cells and grows them in a petri dish, and then exposes them to microplastics and see what changes in those placenta cells. And we have a paper that's under review at Environmental Health Perspectives right now. And what we found was that many of these microplastic particles are taken up into those placenta cells. And we do see very subtle changes in steroid pathways and gene expression. So it does look like these plastic particles are influencing the placenta biology. But again, these are some of the questions that we hope to generate insight into with the Aurora Project.
Mm-hmm. And what about foods? Do microplastics accumulate in certain kinds of foods the way that some environmental chemicals do, like high fat, dairy, meat, fish? Are there certain foods that would accumulate more microplastics?
So there's a few different ways to think of that. There's microplastics that are being generated during the food preparation process, but there's also been a number of environmental studies showing microplastic accumulation in different types of ecological samples. So microplastics have been measured in milk products. They've been measured in a number of different seafood matrices, and this is because microplastics are present very widely in our oceans. So when it comes to accumulating into different types of food, it can be challenging to separate that initial exposure, whether it was occurring to the organism before it became food, or if it is from the way that it's being prepared. So for example, if you are preparing food and storing it in a plastic container, or freezing it in a plastic container and then microwaving it in a plastic container, that's going to generate a lot of microplastics, especially, that freeze, thaw, and heating process. So again, this is one of the areas where we don't know a lot at this point, but we hope to have the methods that will allow us to really understand how these exposures are, or what are some of these sources of the exposure. There was another interesting paper that came out a few years ago showing that plastic tea bags are actually a source of a tremendous amount of microplastics. So there was many that were being formed from the tea bags that could potentially be consumed when you're drinking your tea.
Interesting. And what do we know about the incineration of plastics, especially in low resource countries that use waste burning?
Excellent. Another excellent question. That would be something that we would be very interested in studying. We do have some research projects on e-waste, which is not necessarily all plastic that they're combusting, but they're taking different electronic materials and heating them to extract the precious metals in like circuit boards and other materials. And so that would be an interesting opportunity to look at microplastic exposures from combustion. The issue you face with combustion is it's taking a very complex material, which is the plastic, and heating it and making it even more complex. So there's a lot of different byproducts that can form during combustion, some of which are particles and microplastics, as well as combustion products of those microplastics. So that's a very interesting research question. And this is where the powerful exposome technology that we use really shines, because we couldn't necessarily predict all of the exposures that could be forming from combustion of plastic. But with our untargeted methods, it allows us to detect many of these chemicals and then try and assign identities to them to see what some of the exposures could be.
Interesting. And how can we get rid of these microplastics in our body?
That's another question I wish I had an answer for. And this is, again, something we hope to generate data on. It's not, our intervention study won't answer all of the questions or be able to give you a definitive answer, but it will start allowing us to generate insight and evidence into potential rates of removal of microplastic. I think it's always important to try and minimize your plastic use in general, whether it's from an environmental sustainability perspective or a health perspective. I think that's always a good idea, but plastics are very important to our daily lives, and we all use them most of the day, every day. And so, being intentional and conscious about your plastic use is important, but also recognize that, we don't directly know that these are causing health effects. And, I wouldn't be extreme in reducing your use, but if you can find ways to minimize it and be intentional about it, that's always very helpful.
And what is next for you? What is, do you have any future products? This looks like you're going to be doing this for a long time.
Yeah, so I think this is very much an emerging exposure of interest, and we're trying to help generate some of the key data that will inform on these exposures, and so we can measure them in human populations and in different sample types. My research is focused on using the latest approaches and technology for exposome research to better understand our environment. And so, we will continue to innovate and apply these methods to measure microplastics as well as other exposures that we experience. I have a lot of interest in seeing how these could be linked to cancer outcomes and metabolic disease and inflammation. And so that's where we'll be, focusing some of our efforts as well.
Interesting. Well, thank you, Dr. Walker, and to everyone that joined us today. There is a link into the chat to do an evaluation survey. If you could take a minute to complete that. We'll also email you once the recording is available, and have a great day.